Thank you very much for coming. Thank you for the organizers for inviting me to be here. My pleasure to share with you my research in statistics with applications to genomics data analysis. My talk will be a little bit different from the one you've heard before. It's more focused on statistical methods and where are things that we have to be careful when analyzing biological data. I have two questions in my title, and that will be two parts of the talk, but with most of the emphasis in the first one. Are proteins and mRNA levels related, and why do I care? Why do we care? Okay, so my research interest lies in an interdisciplinary field where I'm merging different concepts from statistics, genomics, and biomedical sciences in general. I highlighted there statistics because that's my field and that's what I mainly do. But I collaborate a lot with people from other fields as well, as many of you here know. The idea is, and my goal is to develop new statistical methods that are needed to study this kind of data. And in particular, I'm trying to make my methods feasible for the analysis of high-dimensional data, because that's the data we usually see in omics studies. And there are three or four main topics that I focus my research on. One is sparsity, and for that I work on regularized regression models where I'm trying to develop new variable selection models, because I'm trying to identify in a large space of features what are the important features that are related with something else, in particular a phenotype. I work a lot in biomarker studies, so I'm trying to identify what are the genes or what are the proteins that are related to a phenotype of interest. And for that I use regularized regression methods. I'm also interested in developing new robust statistical methods, and robust in the meaning of robust to outliers, because in my experience with omics data, I usually encounter outliers in the data that will alter or modify the results that you are trying to estimate. So how can we have methods that are robust to those? And also, how can we diagnose when we have outliers in our data? Because this is a very high-dimensional space, so it might not be as obvious as looking at the data and saying, there's my outliers. Okay, so I'm trying to develop methods to flag outliers and to make the methods robust to those outliers. The data usually contains measurement errors as well, and I just listed measurement errors here, but another one is confounding factors. Both of those problems can be solved using instrumental variables estimators that I will introduce in the second part of my talk. And for the application of instrumental variables estimators in omics data, I needed some relation between different omics data sets, and that's why part of my research was also inclined to looking at the relation and integration of different omics data sets available. So today I will focus mainly on the last two aspects, and if you have questions you can ask me about the previous ones as well. So you probably know more than me about this very complicated space where we have many genes available in an organism. In this particular case, this is a cartoon from the human organism and a human cell. We have many genes, and those genes will be translated and tell the proteins how to function. Proteins by themselves, they may interact and they may form different compounds and have different functions in the organism. So in this space, people were trying to find biomarkers, and that's where I started my research many years ago when I was doing my postdoc. In particular, I was trying to find protein biomarkers for predicting the rejection of a new organ transplanted in patients here in Vancouver Hospital. So I work a lot in proteomics, and I also work in genomics, but this talk will be more about the relation between the genes and the protein. So more about those arrows there. So as you know, mRNA and protein levels should be related, but are also uncoupled by translational rates, different splicing, stability of the mRNA, post-translational modifications, and many other things that can interfere in that relation between the mRNA and the protein levels. So are these two things related or not at all? And that is a very complicated question. It may depend on genes, it may vary gene by gene, and it may depend on the organism that we are looking at as well. But this has motivated many studies today that are looking for the relation between transcriptomics and proteomics in general. One of the questions that they are trying to answer in the literature is, are protein and mRNA levels correlated? And as easy as this question may seem to you, there are many controversies in that level. One of the main questions, and I'm going to focus a lot on that later, is, are we thinking of that correlation across genes, or are we thinking of that correlation within genes? And both are legitimate questions, but they will answer different biological questions as well. So in general, there has been proven very low correlations between protein and mRNA levels, both across and within genes, and that has simulated even more studies on this area. And people are now trying to say, can we predict protein from mRNA levels? So even more than be related. Now they want to say, can we just omit all the proteomics field and just use mRNA? mRNA is much easier to compute and to measure. So can we just use mRNA to predict protein levels? And again, are we asking that question across genes or within genes? And if we're going to predict, how are we going to predict? So most of the studies have used simple linear regressions, and this is kind of the review that I'm going to talk about today, and some problems that I encounter in the literature as well. So as I said before, the first question that we should keep in mind is, are we looking at this question across genes or within genes? And it may seem all very simple to you. I'm presenting it to you as well in another way, but if you go to the literature, it's really a mess. All the studies are mixing these two correlations, are mixing the across genes correlation with the within genes correlation. And when they cite each other's papers, they mix it up as well. So it's very important when you read a paper or when you review a paper to ask yourself, what is this about? Is it about an across gene correlation or a within gene correlation? Because values are going to be different, and the question that you're trying to answer is going to be different as well. So I'm putting as an example here this paper that appeared in Nature 2011 that look at across genes correlations, and you can see a low R² here, so R² being the coefficient of determination and also the square of the correlation. And in this study, we are doing things across genes, so only one sample across all genes. This sample can be a cell line, a tissue, or a patient if you want, but everything across genes, and that's why you have so many dots in that plot. Different from that is to look at this same question within genes. So now we focus on a particular gene. It can be your favorite gene, and you're going to measure that particular gene in many tissues. So here, each dot corresponds to different tissues that has been measured in this study. So one gene, different samples. And I know that some of you might be thinking, this is so simple, what is she talking about? Well, there is a lot of confusion in the literature, and that had created a lot of false results in biology. So that's why I want to emphasize about this as well. There's a very nice review study by Rudy Abersol's group that appeared 2 years ago in Cell, just talking about this confusion, the confusion between across versus within gene correlations, that I encourage you to read. It's a very nice paper, and it tries to state where the field is in terms of are mRNA and proteins related or not. Another thing that I will not talk about, but has been discussed in this study, is where are you measuring this? Is it at steady state or is it a dynamic relation that you're trying to estimate? So the paper that I'm going to talk about today is at steady state. Okay, so it should be the simplest question to answer. Still, there are problems. Okay, so there are two things that are statistically not identical. One is to look at the relation between protein versus mRNA, and for that people have used, as I said, things super simple as correlations, correlation between mRNA and proteins. The results have shown low correlation in general. And they continue, and they were trying to say how much of the protein level variation can be explained by mRNA levels. So when we are talking now about the proportion of the variation explained by another variable, implicitly or explicitly, we are thinking of a simple linear regression model. Simple meaning that there's only one explanatory variable in the model, which is the mRNA. Some of the biologists don't realize that they are doing this. In fact, I'm going to present one of the studies where I have to discuss this with the authors of the paper, and they never assumed that they were doing a regression analysis. And it's true, they never appear regression analysis in their paper, but as soon as you're talking about coefficient of determination, you are thinking of having a regression analysis. Okay. So in this model, simple model that I put here, we have a slope, we have the mRNA, and we have an intercept. In the paper that I'm going to discuss, they didn't put an intercept because they think if there's no mRNA, there won't be any protein, so we don't need the intercept, which is true. But in the data, that is not so true. Sometimes the data doesn't show that, and that causes many problems as well from a data-driven perspective. So if we use, another thing I want to clarify is that if we use ordinary least squares estimation, which is what you most probably will be using, not what all the papers are using, then the square of the correlation is the same as the coefficient of determination. And I want to emphasize that that is true only for least square estimation. It's not true otherwise. And some of these papers are just developing or suggesting a different estimation method and then saying R square is the correlation. And that's not true. R square is not the correlation anymore if you're not using least squares estimation. So it's not only about terminology, but it's also about what your data is telling you. But even more, there's one more problem that is more important than that. There's this paper in Nature as well in 2014, and it's only one section of the paper, it's not the whole paper, that is trying to look at this question. Can we predict protein from mRNA? And after these papers came other papers trying to address the same question. They suggested this idea of using, as I said, a regression model where they multiply the mRNA level by a number, which they mention it's a proxy variable. But that proxy variable has been estimated from the data. That proxy variable is the median of all the ratios between protein and mRNA. As soon as you get that proxy variable from your same data, that is not a proxy variable anymore, that is an estimate. So if it is an estimate, and if you have this form, what you have is a simple linear regression with no intercept, okay? So this puts that full work in terms of where we are statistically speaking, basically. They then predict protein using that estimated translation rate multiplied by mRNA, and they get a predicted value for the protein. So far, so good. There might be differences in terminologies, but there's nothing wrong here. They haven't used least square estimation to predict that slope. They are using the median of these ratios. There's nothing wrong by doing that. In fact, this slope is stronger to outliers than using least square estimation. But as I said before, if this is the way that you're going to estimate your slope, then the square of the correlation is not the coefficient of determination anymore. Okay? So those are things that you should keep in mind, perhaps when reading papers or when reviewing papers or when writing your own papers. Okay, so one slope per gene. So this is what they think is the beauty of their study, that these slopes are gene-specific. They are the translation rate, and that translational rate is gene-specific. So one slope per gene, no intercept. The analysis is done per gene, so one gene at a time, gene-specific slope, and then you can predict what is going to happen. So here, in different colors, I have the different genes. So suppose that this is the slope for the blue gene for gene C, I think it is, and then we are going to use this line to predict. We are going to use this line to predict gene B, and we are going to use this line to predict gene A. Okay? So gene-specific slopes, and we use those lines to predict. Very easy. Okay. Having done that, what they found in the paper is that while mRNA versus protein are not correlated, they have a correlation of 0.4, when we look now at the predicted value versus the observed values, the prediction goes up to 0.9. So they were pretty happy with this because they developed a new method that they can use, and they can predict now protein. mRNA was not correlated with proteins, but now it seems that their predicted values are highly correlated with protein. So based on this observation, and they have found that in the 12 tissues that they have analyzed, they claim that it now becomes possible to predict protein abundances in a given tissue with good accuracy from mRNA. Okay? So this is in that paper. So when I read that paper, I was very surprised because I was not expecting that correlation to be high. And I'm like, how can they go from 0.4 to 0.9 with just a simple linear regression model? There's something weird here. So I decided to get into that data, analyze the data, and see what was happening. So more than that, so this was my question, more than that, if you think what their model is just a linear transformation of mRNA. So the correlation should not change. The correlation should stay the same. So how come they got up to a 0.9 correlation? Okay? So I'm not going to stop here to ask you that question. I will continue. So as I said, we decided to go to their data. These are not the only ones that have done that. Other authors have published very similar results, and the error is getting propagated. So we studied that together with my collaborators, Dr. Paul Pavlidis and Chris Overall, and together with Nick Fortlane as well. He was a Ph.D. at Chris Overall lab. He's now working as an data analyst in Vienna. He finished his Ph.D. here in 2017, and we work on this data set together. So what we decided to do to convince the authors of the paper is to present them. At this point, we already knew what was wrong, but we have to convince them, and they didn't want to believe that their result was wrong. So what we decided to do is we developed 2 control experiments. So in the control experiment, we use exactly their method, just without using mRNA. But we did every other single step in the analysis, we replicated it. We just didn't use mRNA. And then we did the same, just using mRNA from a wrong gene. So we shuffled the genes, and then we used the mRNA from a wrong gene. Other than that, we follow their method. And okay, so this is just a picture of how will you predict with the mRNA method, so without the mRNA. So the mRNA-free, as we call it, basically means that you predict protein with the average protein value, or with a median protein value, and that's what we call an mRNA-free value. The proposed one will be just using the slopes that they propose. When we look at the data that way, we realize that we can also get high correlations. They got 0.9 correlation, we got 0.87 correlation, and 0.84 correlation. Not bad, I'm not using mRNA, okay? So what happened here? What is wrong? So even with this result, we couldn't convince them. We thought that that was something tricky, we were doing something wrong, or anyway, it was a big discussion. So here is the mRNA versus protein level, the original one, here is the proposed one, and our controls. So something is wrong here. And as I said, this is very simple statistics, right? Simple linear regression, correlation, nothing very tricky. So what is going on? Okay, so what's going on is that when you want to evaluate that prediction method, again, there are two ways of evaluating the prediction method, across genes or within genes, and that makes a big difference. So because we have predicted within genes, all that we are doing is really a rescaling of those mRNA values. So here the open dots are for the mRNA values, and when you predict using that transformation, the transformation starts multiplying it by a scalar. What you're doing is moving those open values towards the 45-degree lines, okay? And it's the 45-degree lines because here I have predicted versus protein. So of course the predicted value will look similar to the original protein value, right? And so they will move towards that 45-degree line. And I do here, this is a cartoon, but it's based on real data, I think. This is the turquoise gene, the purple gene, the red gene, the green gene. In all of them, it's happening the same phenomenon. The mRNA is getting towards the 45-degree lines. Okay, so now I have to evaluate my predicted values. Now I have to evaluate the solid dots, okay, to know how good that prediction was. In other words, how tight are those solid dots to the 45-degree lines? If they are closer to the 45-degree lines, then I have a good prediction, because in the 45-degree lines, predicted is the same as observed. If they are not close to that 45-degree lines, then my prediction is not good. Okay, so not that difficult. Okay, so here with ellipsoids, I'm illustrating the correlation or the relation between the predicted values and the observed. If you look at those ellipsoids, they are not that tight, okay? So in particular, this one, it's not that close to that 45-degree line. Also, and the same happens perhaps with others. Some of them might be better than others. Okay, but this is not what the authors have done in that paper. What they have done is, okay, so if I look at the data that way, I will get what I was expecting. The correlation is the same between the original mRNA or the predictions that I get, because my predictions are just a linear transformation of the original data. So I get exactly where I started with. There's nothing new here. The correlations are low. They are centered around 0.2, as it was expected, and it was always said in the literature. Some are even negative correlations. Perhaps they are not significant correlations, but they are negative. As I said, not having an intercept is sometimes a problem. And this is just an illustration of one of the genes. So when I look at that question within gene, across the samples, then I don't have a good prediction method. So what has the author done to get that 0.9? Well, what they've done is that they predicted within gene, but then they evaluated across genes. So they took, let's say here, one kidney gene turquoise in kidney, and then the other gene in kidney, and another gene in kidney, and another gene in kidney. So if I now look across genes for a given tissue, then the correlation is large, okay? And imagine I'm doing this just with 4 genes, but imagine having many of the genes and doing that, because of the high dynamic range that you have in protein values, then these predictions start getting larger and larger and larger. So if you look, because the within variation of genes is much smaller than the across gene variation, then you get very high correlations when you look at this problem across genes instead of within genes. Okay? Is there any question there? Yeah.
two plots. One was predicted, one was
This one?
RNA over fat. The data is the same on both those plots, but with different colors, but it's different quantity on them.
Yeah, so here, so in open is the mRNA, and these are the predicted, and so the solid dots here are the same as the solid dots there.
Yeah,
They don't look the same?
they look exactly the same.
Yeah, they are the same.
But then one is predicted protein.
Yeah.
One is mRNA.
I have both here. I have the mRNA in open and the predicted in closed. And here I'm just trying to simplify the figure and just put the solid dots. Okay? So what I'm trying to show in this figure is how you go from the original mRNA to your predicted values. So what that transformation is doing, what the method is doing, is just moving dots towards the 45-degree line. That's all that their method is doing. In this, I want to show you something more simple, just the predictions, so just the solid dots. So that's why if you just focus on the solid dots, the two figures are the same. Okay? Now just looking at the predictions versus the observed, are they tied to the 45-degree lines, yes or no? So that's why I show you here the ellipsoids to tell you that sometimes they are tied, sometimes they are not so tied. Okay? Okay, so well, the prediction can go both ways depending on where the point is. Yeah. Overall, they are shrinking towards the 45-degree lines, but it's not that they always go there. Yeah. 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 That's a good point. Okay. So I was here before. Okay. So as I made a comment before, if you think of correlations being something that forms a line, if you spread out the points, because the protein values have that large dynamic range, what you're doing here is spreading out the points along a line. And the more you spread them, the more correlation you will gain. Okay? So this axis of that ellipsoid will gain over that axis of the ellipsoid. So we wrote an article responding to this, highlighting the problem and trying to respond to this problem. And it was published last year in Nature after many conversations and revisions, but it was finally published. There's some references there in the paper towards more geometrical interpretations of the problem. So these things that I'm saying about the ellipsoids, there's a very nice paper that if you're interested, you can refer to it. But correlations are usually visualized with these ellipsoids, so you can use them to visualize your data. And the more you spread out the points, the higher the correlation will be. So by just taking only per tissue analysis across all genes, they created that high correlation. But I did the same. I didn't use mRNA, and I created the same high correlations, right? Because I also have the high dynamic range of the proteins. So even if I use, my ellipses are going to be worse perhaps, so per genes my predictions are going to be worse. But when I look across genes, I have very good correlations as well, just as they have, okay? This problem is well known in statistics. It's usually referred as the Simpsons Paradox, not in relation to predicted versus observed, more in relation to data. Sometimes you have some relations within groups, but if you ignore the groups and you look at the data overall, you see another trend. So here, for example, we see a positive relation between the blue dots and the red dots. But if you ignore that there are blue dots and red dots and you look at just what is the correlation in the data, the correlations appears to be negative. In our case, what is happening is that if we look at the correlation within the genes, okay, between the predicted and the observed values, we have a result. But if we look at that across the genes, that result changed. So it's very related to this paradox. Okay? Yeah.
So
So what I'm saying is that the analysis should happen within genes. For some genes, there will be good correlation. So these ellipses are also correlations. For some genes, there are going to be good correlations. For other genes, they might not be good correlation. The analysis should be done per gene and not across genes. You can do it across genes as well, but then the predictions has to be across genes as well. What you cannot do is to mix match. You cannot do a prediction within and an evaluation across. Okay, that's the main message of the paper. Okay, so for those that are more interested in statistics, what we are really doing here is fitting a mixed effect model, where we have a random effect here, a random effect for genes. If some of you have ever heard about mixed effect models, otherwise just disregard this slide. So this is a random effect for genes. This will be the slope, and we can even think of these two being interacting here, and then we will create a random specific, a gene-specific slope, okay? If you do the analysis that way and you want to know if mRNA is significant or not, what you should do is you should compare a model with mRNA versus a model without mRNA, exactly as we did in our control. In fact, we did our control because we knew this, but we did it in a visualization way. What you are doing statistically is comparing the mean square error, not the correlations. I don't know why biologists like correlations so much. The mean square error is a better measure because it's not affected by that linearity in the data. It's just a distance. So now we are looking at how far my predicted value is from my true value, not how far my predicted value is related to my true value. So if you look at the mean square error now, which is how far the prediction is from the true, and this is the Euclidean distance of that, then you can see that in half of the genes, or about half of the gene, the mRNA-free method is closer to the true value than the prediction that you get with this translation that they suggested. So when you get these results, then half of the genes, one method is better, half of the gene, the other method is better, is when your test is not going to cause significance. Okay? So if you do a proper statistical test, looking across genes now, but properly accounting for that within gene estimation, mRNA is not significant. That part is not in the paper. They didn't want the paper to become too technical. So that part is not in the paper. But if you analyze the data that way, you will see that mRNA is not a significant predictor. The gene will be a significant predictor. But what this gene is capturing is that high dynamic range variation in protein levels. Okay, so if you're interested in playing with this data set, we have created a Shinyup. My student, David Kaplinger, helped with that part, although he's not an author of the paper. He worked on the Shinyup. In this Shinyup, you can select how many observations you want per gene, because sometimes the gene was not measured in all the tissues. So you can say, I want only the genes that has been measured in most of the tissue to make part of the analysis, and you can see what is the histogram of the correlation, so you can play with that. You can also look individually at genes. There's a drop-off window where you can select your gene of interest, so you can play with your gene if you want. One of the scatter plots will show you the original mRNA versus protein levels. The other one will show you the predicted value versus the original values. You can also look at those ratios, because the authors in the paper claim that those ratios were constant. So by looking at the plot, you can have your own idea if they were constant ratios or not. Okay, so all that can be played with the data. So just to conclude on this first part of my talk, when you're looking at correlations between predicted value and true values, you have to be careful if that evaluation is done across genes or within genes. The effects can be very large, but mainly driven by the high dynamic range in protein levels. And we have proved that using 2 control experiments. The within gene predictions must be evaluated with within gene correlations as well. If you want to use correlations, if you want to use mean squared errors, then it's a different story. Mixing across and within gene analysis, it's not a good idea and can lead to misleading results, as you have seen. A full analysis of this data and also a full analysis of EdForce data has been published almost together with our manuscript in Nature in PLOS Computational Biology. The authors have done a beautiful job analyzing these 2 data sets, so for those that are interested in these questions, I suggest you to look at this paper as well. And at least in this data set, we haven't seen very strong relations between mRNA and proteins to claim that we can predict protein from mRNA. There are new studies coming. I have seen they're published, and I'm also reviewing papers from people looking at single-cell data now and looking at this question at the single-cell level. It is still an open question. It's still an interesting question. What I suggest to you, when you are a reviewer or when you are a reader, pay attention. Are we looking at the question within gene, or are we looking at the question across gene? Are these 2 being mixed up? Because that can change a lot the results that you look at. Okay, having said that, I have 10 minutes to go to the second part of my talk now. Why do I care? Why did I start looking at this relation between mRNA and protein? And I know it's an interesting point, and I know there's a whole field looking at that question. But in particular, me, why did I care? And it's because I was interested in biomarker studies, and I work a lot in that area. And when I work in biomarker studies, what I do is I model the relation between potential markers and phenotypes using a linear model. So this is my equation of a linear model. Now the problem is that the least square estimator, or any variation of the least square estimator, lasso, or other regularized methods, will give me biased and inconsistent results. More the inconsistent is the problem. Bias we can accept for many reasons, but also inconsistent results when there are measurement errors in the covariates, or when there are confounding factors or bias in the sampling. What I mean by inconsistent is that your estimator is always going to be biased, no matter how large your sample size is. That's what I mean by inconsistent here. There's a technique in statistics called instrumental variables estimation. In fact, it came mostly from economics, that provides a solution to this problem. So borrowing that idea, I'm going to show you here a very simple example of a confounding factor. So when we are looking at the relation between a feature X and a response Y, sometimes we have these confounding factors. Sometimes we measure the confounding factors, sometimes we do not measure the confounding factors. A confounding factor is a variable that affects both the explanatory variable and the response. And that creates a spurious relation here, because now I don't know if X affects Y directly, or if X affects Y because it's affected by U. And there's this backdoor path that can create that spurious relation between the variables. And that creates the bias. So how can I destroy this path in a certain way, or how can I overcome that path? The idea here is to have what is called instrumental variables. So these instrumental variables are my special helpers. They are other variables that has to be associated with X, and I'm going to put them back in the diagram here now. If I can find a variable that is associated with X, and only with X, and is not associated with Y otherwise, so it comes to X only to Y only through X, then that is the definition of an instrumental variable. Why this is a good definition of instrumental variable? Because X is a problem here. X is affected by U, and I want to have something that do not go through U. So the idea is to go in 2 stages. In the first stage, you project X onto the space of W. And by doing that, I'm not going to use X, I'm going to use something that lives in W. And W is not related with U. So if I use W or something related to W, I overcome this problem. So the first step is to regress X onto W and to get X hat, the prediction of X in the space of W. In the second stage, I'm going to regress Y on X hat, because X hat is really a function of W, and W doesn't have any problem. So by doing this two-step estimation, I overcome the confounding factor. The same is true for measurement errors. I work more with measurement errors, but the picture doesn't come so nicely, so I decided to show you the confounding factors. And using this technique, we can solve the inconsistency that comes through measurement errors, confounding factors, or dual causality. Okay, so how am I going to use this technique in biology? I'm interested in this. I'm interested in finding a relation between proteins and a phenotype. For example, the stenosis in a bulb. I'm trying to find by biomarkers. But if the relation I'm estimating is biased, I may miss some biomarkers. I may miss some good biomarkers, and I may catch some false biomarkers just because of that bias estimation. So how can I overcome that bias? Well, the idea is to use instrumental variables. But for instrumental variables, I need an instrument. Well, the instrument I'm going to find is this relation that I have before the proteins. So I can use the relation between the mRNA and the proteins to create that W. So this will be my W that is related to protein and then to the phenotype. Okay? So I will use the predicted protein values instead of the true protein values to relate it to stenosis in this example. Okay? But in order to do that, I need this relation to be true. I need the mRNA to be related to protein. So that brings me back to my first question at the beginning of my talk. Are they related? Can I use them? And that's how I studied that research topic. Even more than that, we don't have only one gene. We have many genes, and these genes are related. Should I use just one gene for each protein, or should I try to select from all the networks of genes what are the ones that are relating to my protein of interest? Plus, I don't have a protein of interest. I have many proteins, and I also have to select. So that brings regularization into place. And that's how all the things are connected that I mentioned in my first slide. I'm interested in regularized methods. I'm interested in robustness that I haven't shown you today. And I'm interested in the relations between mRNA and proteins because I need them for instrumental variables models. So with that, I want to thank my collaborators and co-authors of the Nature paper. Dr. Nick Fortlane, as I said, was a student in Chris Overall Lab. Now he's working in Vienna. And Paul Pavlidis was a great contributor as well of the project. And David Keplinger in my lab working on the Shinyup. With that, I will thank you and open the floor for questions. Yes.
I have two questions. So first question is, so you have looked at the advantage of the coding method. Now, what if you also add other dimensions about, say, copy
Yeah.
like getting the loss? How would you model that?
That's a very good point. And in fact, sorry, do you want to finish?
Yeah. The second question is about some of the published papers, including some of the market analysis, showing that the discordance between mRNA protein expressions are most likely due to, say, those proteins which form part of the multivalent complex tend to show mRNA protein dysregulation. And some groups have hypothesized that there might be some compensatory effect going on this multivalent complex. So have you thought of associating functional dimension to the
Yeah, that's a very interesting point. So going to the first part of your question, should we look at other parts of the genome, perhaps looking at mutations or looking at more at the genetic level? There are many studies not many studies, but the first kind of studies that started working on this are called Mendelian randomization. So if you're interested in this, there's a whole literature called Mendelian randomization, where more genetic variants are trying to be used as instruments instead of mRNA levels. And at the end of the day, they might be a better idea than trying to use mRNA levels, because one of the problems with mRNA is that, for example, in the case of confounding factors, we don't know if the confounding will not affect the mRNA as well. The variant can be argued that was given randomly at the meiosis stage, but the mRNA expression can be affected by the same confounding factors as the protein expression. So if you're trying to address confounding factors, using mRNA expression is not a good idea, and you should go to other parts. Okay, you can complement, you can use both. You can search that the space gets larger, but you can combine more than one omic. You don't have to use only one. And it might be a good idea to combine them. They might be related. So when you look for a regularized method or for a network, you may have to take that correlation into account. Okay? So yes, those are very good points. Going to your second question, the relation between protein and mRNA, so that research is not closed. It's an open question, and I agree with you. There are other studies that look at this problem in a more comprehensive way and trying to explain what are the different sources that explain why the protein level is far from that mRNA level. So far, I didn't show you that. I just show you that we don't have a good correlation. Okay. Now, why? So those kind of papers are trying to address where is all that information falling. Okay, so yes, it's a very interesting field and literature. I just wanted to point out here, mostly to make you all conscious of this problem in the literature. Are we looking at the problem across genes or within genes? Because it's a very subtle thing, but it creates an enormous impact, as you can see. Okay? Yeah. There was another question.
What would be an example of a confounding factor in this where mRNAs are X and proteins? What's an example of what you might use?
Yeah, so for example, even here, smoking can be a confounding factor, right? Like if the patient has been smoking, it can alter the stenosis in those valves, and it can affect the protein, some protein levels, for example. So smoking status can be one. Perhaps you measure smoking status in your patients and you can put it into the model, or perhaps not. Or it can be exposure to something that perhaps it's about workers working in a field, in a particular field, or working with, I have one of my collaborators, they are looking at asthma reactions in workers working with red cedars, and they become asthmatic and allergic to the red cedar, so the exposure to that, to some things that they don't know exactly what they are, so they can measure it. So those are examples of confounding factors. Sometimes it's even socioeconomic variables that you cannot measure them all or put them all into your model as well. They can affect protein levels as well as the state of a given disease. Yes?
So the upshot, if I understand it, is that the previous analysis by the previous group,
Welcome.
They would make a prediction for protein expression level, and it would look great on the curve, but it would be incorrect if you actually went to measure it. Whereas, I guess the conclusion from your study is if you went in and measured it, you wouldn't be able to predict that. They would find data that would be off their curve. And I guess my question is, can one actually just falsify it by taking an mRNA expression level, looking at their prediction, what the protein expression would be, measuring it, seeing by qPCR or something, and just falsify it?
Yeah, there can be some additional experiments to try to validate those results. What I just want to clarify is that there's nothing wrong with their prediction. It's just very naive. It's just too simple to claim that we can predict. So they have done nothing wrong in trying to predict it that way. At the end of the day, it's a prediction, okay? But it doesn't work. What was wrong was the way that they used to justify that it works. It doesn't work. And it doesn't work because it doesn't work for each particular gene. When you look at one gene in particular, you see that it doesn't work. The results that they got, it was an artifact of looking across the genes. There's nothing wrong with the prediction itself. What is wrong is the way that they evaluate their prediction method. Okay. Yeah. Even if they tested with another method, if you look at cross genes, you will find those high correlations again. So it's more using the statistical tools that are needed. Yeah. Yeah.
So I guess I'm more of a biologist, so naive thinking here, but wouldn't another interpretation be that the protein abundance alone, like whatever range of that dynamic range of the protein abundance, will kind of predict the range of like you would expect the mRNA to be in, and therefore, like, yeah, I understand that they're moving everything onto a diagonal, doing that regression, but isn't that still improving your ability to predict how much protein there would be from mRNA? Because you're just accounting for the differences in the dynamic range.
So it predicts something. So you're predicting something. So I read another paper where they are now comparing with the mRNA-free, and they're saying we are better than the mRNA-free. Of course you're better than the mRNA-free. So you are using some knowledge, right? So the mRNA is giving something. The question here is it giving enough? Is it significant enough to say that to claim that you can predict? And what is happening is that it goes on gene by gene basis. For some genes, it's enough. For some genes, you can predict very well. For some other genes, you cannot predict very well. What you cannot do is to try to answer across genes. Okay?
So were they using one R value for all genes, or every gene had its own R value?
Every gene should have their own R value. And that will be the correct way.
Yeah.
What they did was a hybrid because they used one gene per tissue. And so statistically correct, you either have one R value per gene or one R value for a mixed effect model. What you cannot have is one R value per tissue. There is where you broke down things into a weird way and you create an artificial high number. Okay? Now, when you started talking, you said something, and perhaps this was not your point, but you remind me something, which is all this high correlation is basically artificially inflated by the large dynamic range. So what happens if I destroy that large dynamic range? And I can destroy it by just normalizing the data. And if you normalize the data, results are gone. You don't have that 0.9 anymore, okay? So, yeah, so that's another way of proving them wrong. But they didn't want us to get so technical in the paper, so we took that part out.
But, devil's advocate here, but my point would be that the whole point that, like, the dynamic range differences, that is helping you. Like, that's important.
That is help. Oh, that helps in general. Yeah.
Like,
it doesn't help the within gene. It doesn't help the within gene. It doesn't help the within gene. Yeah. Perhaps there is some genes that has also more. So okay, I know what you're asking now. So some genes, the prediction works well, and it works well depending on how much variation they have. That's true. Yeah. The method will work better if you can capture some variation. I think that's what you're trying to say. Yes. What you cannot do is generalize that for every gene this is going to work. So it works for some genes. If you look at the histogram, some genes are doing really well. Yeah, if you look at the histogram, some genes have very high correlation here, right? So now which are these genes? So if you go to that paper, this paper, they have done a more extensive analysis. So they have looked at what are the genes where that correlation is good. Is there a do they belong to a particular family? Do they have more variation in their values, and that's why the method is working better? Yeah, so I understand now what you're saying, yes.
Do you think that would be reproducible? Like those genes that worked well, if you just redid this experiment, regenerated the data, would you get a different set of genes that worked well?
I don't remember what these authors said, but because they have done, I think, 3 different independent data sets, they may address that question as well. I don't think they are reproducible, honestly. These are based on 9 points. So how reproducible that can be, right? I believe more on the single cell now, because if you now have many multiple cells, then that can be different, right? Yeah. Fine. Yes.
Okay. So the protein level is like, so if you think of mRNA as a function over time, and then protein as a function over time, for just one gene, right? And the protein over time is an integration of the mRNA over time, right? Like that you had one transcript, and let's say it was translated once an hour every hour for this gene, right? And that transcript would, then after one day you would have 24 copies of that protein, right? So it was translated once every hour, 24 hours. So, but then we look at a snapshot of time of this function, right? Where how important is that this variable is an integration of that variable?
I don't know if I'm going to answer to your question, but the analysis here is at steady state, so no time, no dynamic in time.
Okay.
But there have been people that try to model that dynamic in time using differential equations, and they have found different results now. And I've seen papers also that they see that sometimes the response of the protein is coming later, but it's coming. So if you look at a particular time, you don't see that relation. But if you allow that time to progress, then you see the relation better between protein and mRNA. So I'm not trying to say that protein and mRNA are not related. I think they should be, and we should understand it better. But yeah. But not by that linear regression. We have to think harder than that. Yeah, the organism is much more complicated. Yes?
So what do you think is driving the difference between mRNA and protein levels? Do you think it's due to epigenetic effects that are affecting some sets of genes? So you can get a range of genes that are not affected by epigenetics that maybe then have good correlation, whereas ones with high effects maybe less so?
I think it really depends on the genes. Some genes will be affecting more than others about factors like ethnicity, exposure, as I said before, to drugs, to smoking, to environment. So genes will differ. That answer will differ gene by gene, in my opinion. I'm not an expert on that also. So, yeah. But that's why I found it more interesting to look at this question within genes than across genes overall. But there are many studies across genes that are perfectly fine. What you cannot do is to mix them. I hope I passed that message. Good. Well, thank you very much.